Hi, everybody. Thank you so much for coming. We are really happy to have you all here. It is 10 o'clock Pacific time, 1 p.m. Eastern time, whatever time zone you are in. Thank you so much for coming. We have our CSO, Giovanna Scapin, with us, and I am going to hand it over to her. Just a quick reminder, at the end, we will be taking questions. At the bottom of your bar, there is a little box that says Q&A. So you can ask your questions live there, and Giovanna will be available to answer. We will be sending out a recording of this at the end, so you will be able to rewatch. And if you want to email us any questions afterwards, our email is info@nanoimagingservices.com. Or you can contact us through our website at nanoimagingservices.com. And I'm going to go ahead and hand it over to Giovanna now. Thank you all so much for being here.
Good morning, good afternoon, good evening. I don't know where everybody is. Thank you again for joining this webinar. I'm really very happy to be here with you to discuss, talk about epitope and paratope characterization using cryo-EM and the new service that nanoimaging services is offering in this area. The webinar will be mostly about epitope mapping, but I will start with a brief introduction about cryo-EM and the issues, the problems, the challenges that we often face, so that proper expectations can be set. You know what you're expecting and we know what we can offer you, and that the requirements that I will be briefly touch upon later can be fully understood. A little bit about NIS. We were founded in 2007 by Bridget Carragher and Clint Paller, whom I'm sure most of you are pretty familiar with. On the bottom of the slide, we can see a brief summary of the timeline of our companies. We now have 3 sites, 2 in San Diego and 1 in Woburn, just on the outskirts of Boston. We are a fully private company. We own 7 microscopes, and we are dedicated to all things cryo-EM, from single particle analysis to nanoparticle characterization. Because of the scope of this seminar, I will not touch upon characterization at all. But as Chloe said before, if you are interested in knowing more, you can either reach out to us to our website or the email, or you can just look to our website for all the information that we have up there. I will focus on single particle analysis because that's what we are going to use to do epitope mapping. And I am just going to spend a few minutes describing the experiment. Now, I don't know the expertise of the people attending the webinar, so I apologize in advance if what I say seems very basic, but I think that sometimes starting from the basics is very important to understand the full process. So in 99.9% of the cases, your sample will come in a little tube, and all the protein in there will be happily tumbling around in the buffer of choice. The first and the most difficult step in the process is vitrification, during which we take a small aliquot of this sample and we apply it to a grid, and then we plunge the grid into liquid ethane, so to vitrify it. And the goal is to obtain a thin, uniform layer of ice in which the sample is embedded and oriented in many different ways. We do need this distribution of orientation because what we actually measure when we send the electron beam to our sample, to our grid, are 2D projection of these particles. And we want to be able to capture as many of these views as possible so that we can put them together in a 3-dimensional reconstruction. The signal-to-noise ratio of the images that we collect, which are called micrographs, it's very, very, very low. And in order to use the information in the micrograph to get something that we can work with, we need actually to be able to identify, pick, classify, and align all of these particles, these low signal objects, in a process that's called 2D classification. The result of this process are 2D classes in which we have all these different orientations, or as many orientations as we can possibly have, and in which the signal to noise is highly enhanced, and we can start seeing details, secondary structure details, or details of the particles. These 2D classes are then used to generate a 3-dimensional reconstruction. The most important thing to remember is that we talk about single particle analysis because we look at individual particles, antibodies in our case, antibody-antigen complex in ice. But what we are really working with is always an average of many of these individual projection images. And now we are thinking about averaging. If we want to get something out of this micrograph, out of our entire experiment, we need to be able to pick this particle correctly in the process that we call picking, properly group them accordingly to shape and orientation, and properly align them first into two dimension and then in a three-dimensional space. And that's where some of the limitation of cryo-EM come from. In order to be able to carry out steps 1 and 2, sorry, we or the computer need to see the particles and be able to identify them and not just see them, but also be able to classify and group them into classes. And in my view, this is the biggest limitation of these steps comes from the size of the samples that we are looking at. There is a reason why we have lots and lots of maps for systems that are 100 kilodalton or bigger, but very few for systems that are smaller than that. And it's because small particles are actually very, very difficult to see, very difficult to orient, very difficult to classify. In order to be able to properly align things, so step number 3, we also need to have as much order and or conformation and stability as possible. The better we align the particles in 2 dimensions and then in 3 dimensions, the more details we are able to extract from the micrograph, the higher the resolution of the system that we can see at. What we know is that flexible things like this dancing antibody doesn't really work very well in cryo-EM. This brings up the concept of requirements. What do we need our sample to be like in order to get a successful cryo-EM experiment? And we still ask for a routine 3D reconstruction. We prefer the particles that have an ordered mass greater than 80 to 100 kilodalton. We can do smaller particles. We can do smaller, more flexible particles. It just won't be a routine experiment. And that we are talking about something that becomes routine in our structure determination process. And again, won't be routine. We can do it. It will require much more time and much more money and many more resources. As for amount of sample that we ask for, about 50 to 100 microliters at about 0.5 to 5 mg per ml are enough. If the antigen is a membrane protein, the ideal concentration is about 2 to 5-fold higher. The buffer is the buffer of choice. It's the buffer in which your sample is the happiest, in which is monodisperse, in which is soluble, in which we can get to the concentration usable for cryo-EM. Cryo-EM is fairly tolerant of buffer composition. Basically, anything goes as long as there are no components that will interfere with the vitrification process or with the signal-to-noise ratio. So compounds that have a high carbon contents are not preferred, like glycerol or sucrose, or organic solvent that like DMSO that do interfere with the vitrification process. Okay, let's try to keep this in mind because this is important, will be important at the end of the webinar. Let's talk about the topic of this webinar. So if you're here is because you probably know already how important it is for biologics discovery and development to be able to identify and characterize the areas of interactions between an antibody and its antigen. Reasons go from like basic research, how do they bind, to all the way to patent protection. And over the year, many different techniques have been developed and improved to characterize this interface. These are just a few of the examples, and depending on the question we have, and why not, on the time we have and the money that we have, we can select one or more of all of these different techniques. They do not provide all the same answers. That's why I'm saying depending on the question we have. And the details they provide can be anywhere from just the linear sequence of amino acids involved in the interface to actually a real description of the spatial arrangement of the different components. And as we all know, we are in a more and more competitive landscape. And it's becoming more and more evident that the need for increased detailed information as specific characteristics of our biologic entities and how they interact with the intended target is rapidly growing. That's why we are getting more and more interested in obtaining atomic level details that will allow us to understand the specific interactions at the interface and will help us elucidate, for example, the mode of action, or will help us in designing optimized interface with increased potency or specificity. Structural or three-dimensional epitope mapping can straighten patent claims by providing detailed information about unique aspects of an antibody binding to the antigen, can demonstrate, for example, novelty, specificity, and utility of this novel antibody, increasing the chances of successful patent applications. As typical is, there are different techniques that we can use. A very commonly used one is the hydrogen deuterium exchange coupled with mass spectrometry. In this experiment, we selectively deuterate specific residues, and by comparing the mass of the complex, the antibody-antigen complex, with the mass of the individual components, the antibody and the antigen, we can identify potential peptides that are involved in the interface, in the antibody-antigen binding. I labeled this technique as a low resolution because it actually provides, I will call it peptide resolution level data, and it really doesn't provide any quantitative analysis and identification of the specific residue-residue interactions. But if the structure of the target is available, for example, and we can build a model for the antibody, the data can be then mapped on the surface of these models, and modeling can be further used to derive atomic level interactions. The same kind of information can be used if we use negative staining electron microscopy, in which we can provide nanometer resolution envelopes of the systems into which antibody and antigen can be then docked and used for initial interface determination. But if we want, what we need are real atomic details, then we need to use what we call high resolution techniques, which are basically X-ray or cryo-EM, which they provide us with a three-dimensional structure of the complex and the atomic level details we are looking for. One way the knowledge of the atomic details of the epitope paratope interface can help is, for example, during the process of affinity enhancement, using structure-based and computational methods. A starting antibody may be obtained via de novo design of a pool of pre-existing antibodies. 3D, so atomic information, can then be obtained by either experimental methods or purely computationally approach, or a mixture of the two. Computational approaches, due to the recent breakthrough in computational structure prediction, have become more and more accurate and have been using more and more often in order to help this entire process. The three-dimensional information, in combination with a large, rapidly increasing numbers of available sequence of antibodies, will allow for like a large-scale structural antibody virtual screening, and this can be then followed by silico mutagenesis, experimental and prediction, in silico prediction, experimental assessment of stability, and the process can be repeated over and over again until the final or a suitable antibody construct has been designed. And any technique can be used at this point. Can be used X-ray, can be used cryo-EM, or, as I mentioned before, HDX. I would like to give you a little bit of an example of the complementarity and differences, though, that exist between different techniques. And I'm focusing on HDX, which is very commonly used, and three-dimensional structure determination. This patent highlights the diversity between three different antibodies that are labeled 1, 2, and 3 against the same target. And both hydrogen deuterium exchange data and cryo-EM structural data were generated in support of the patent claims. The idea is to prove that these antibodies are actually different. And so if we look at the HDX profile of the antibody 1 and antibody 3, they actually, so this is the antigen HDX profile in presence of antibody 1 and antibody 3. And they're actually different, right? We can clearly say that the profile is different, that the antibodies will likely bind in two different regions. And so antibody 1 and antibody 3 are actually two different entities from any scope and purpose. And indeed, when the structure of the cryo-EM structure of the complex between the target and either one of the two antibodies was solved, it was clear they did bind in different orientation. The target is the dimer in the middle, and the two antibodies, a light gray and a dark gray, will bind in two different orientation to the antibody, to the antigen. So HDX already had told us, in this case, it was just confirmed by cryo-EM. But the HDX profiles for what we call antibody 1 and antibody 2 are basically identical. And in this case, just relying on HDX data, we can't see that these are the same thing or two different things, or they have actually enough differences to warrant a separation on activity of the two antibodies. In this case, the generation of three-dimensional data, three-dimensional structure that was done with cryo-EM is essential to assess atomic differences. Like in this case, the different position between the CDR2 loop in the two antibodies and the different interaction with the antigen that are generated by a local in the antibody change of a phenylalanine to a glycine. So I think that we all agree that having three-dimensional structures is very relevant. But how do we go about it? The two major techniques are, of course, X-rays and cryo-EM. But how do we pick the most suitable? I tried to write down some of the real or perceived pros and cons of both techniques. Of course, if the complex of interest crystallizes, go for it. It's likely a faster, cheaper process, resulting in a much higher resolution data. But sometimes the targets we are working with are not amenable to crystallization. Or maybe the recombinant Fabs are not available. We only have the full-length Fab, the full-length antibody, and we may or may not able to carry out digestions. At this point, cryo-EM seems to be the right solution, but cryo-EM is notoriously slow and expensive. Or is it? Well, in the past 2 years or so at nanoimaging services, we have seen almost 30 epitope mapping projects. And they all end up with a resolution at the interface or around 3 angstroms, or even better. Definitely at a resolution that was always suitable for chain tracing and side chain assignment. Although most of the projects ended up on the cryos because they followed our standard protocol of screening and optimization of the grids on the glaciers, and then final data collection on the cryos, a retrospective analysis of all the available data showed that for many of these complexes, the glaciers data were actually already enough to get the desired information. Maybe the overall resolution was a few tenths of an angstrom lower than the one that we obtained on the cryos, and the local resolution was maybe a bit lower than what we obtained on the cryo, but in any event, it was always good enough for chain tracing and side chain assignment. So based on this analysis and considering that when we start an epitope mapping project, the question is, what is the epitope paratope interface? And likely not, what is the overall structure of the protein I'm looking at? We conclude that cryo-EM can indeed be very suited for providing answer, what is my epitope paratope interface? With a good resolution, definitely able to trace the side chains at the interface, can be really fast, less than 2 weeks turnaround, and at overall cost that is much lower than what is usually considered the normal cost for a full 3D map reconstruction. And of course, the system, we can use the same system for any of the various next generation antibodies, right? From the nanobodies all the way to this Z-body or whatever we want, as long as the size and conformational stability of the complex meet those minimum requirements that I talked about. So size around 80 to 100 kilodalton, conformational stability, and the conformational stability. I just want to briefly spend a couple of minutes why you guys would like to work with us to do your epitope mapping needs. And as I mentioned at the beginning, we are a purely cryo-EM company, which means that we really know how to do it. As I said before, we own our microscopes. So what we can assure is fast and reliable access. We have a staff made up of very experienced microscopists and PhD level scientists. We are very collaborative and capable of engaging with you at every step of the way. We have many flexible services and access modes that can be tailored to your specific needs. And this epitope mapping package is just an example of that. And most importantly, or not, last but not least, we take data security and confidentiality very, very seriously. And with this, I would like to thank you all for attending, and I will happy to answer any questions. Okay, so I see there are a couple of questions on the Q&A. Maybe I'll stop sharing the screen. So the first question is there a size limit, upper limit and lower limit for the antigens? No, but the lower limit is that the antigen-antibody complex or Fab complex has to be at least 80 kilodalton. There is really no upper size limit. We work with megadalton proteins, and that works well. Is the lower limit that might be a problem. How often do you need to optimize the conditions for grid freezing? Are the conditions strongly antigen dependent? We need to optimize the condition every time. So every time a new complex comes in the door, we consider it a new sample, something that we have never seen before, because even a different constructs can behave differently in our hands. And a construct, the same construct coming in with two different buffers might behave differently in vitrification. So we do screen all the time. The idea is that once we identify conditions that are suitable, they can probably be reproduced on success on the next experiments if the only thing that changes is the antibody, the Fab, and not the antigen. The conditions are strongly antigen dependent. Usually the position in which how the antibody binds to the antigen can also be affecting not as much as the vitrification, but the final results, the way the particle looks so nice. So there are both things that we need to control or keep an eye on. Do you find that glycosylation is a problem for cryo-EM and antibody complexes? Is the deglycosylation required? No. Glycosylation is not a problem in cryo-EM at all. Since we are looking at the individual particles, we don't need to pack them. Even entities like glycans that are disordered don't really interfere. The end result is that we will not be able to see them in our micrograph or in our final reconstructions, but they definitely don't affect the final reconstruction at all. And so no, glycosylation is not necessary. We can take natural proteins, natural systems, and just work directly with them. It's a KD criteria that needs to be taken into consideration for a project. Thank you, Liz. This is a very good question, and the answer is yes. Cryo-EM works a concentration that are micromolar. So the 0.5 to 5 mg per ml is approximately on the micromolar level. And so anything that has a KD worse than micromolar will probably tend to dissociate. And that is our kind of guideline. We have a way to address that. We can work, if the KD is worse than the micromolar, we can address it by maybe using a higher concentration of one of the two components, or we can start out doing the incubation at concentration that are much higher and then dilute them immediately before preparing the grids. Or we have a chameleon in Woburn near Boston. The chameleon is a different vitrification device that allow us to use protein concentration much higher than the one that are normally used for the vitrobot. And so we can address the lower affinity using one of those instruments or other similar ways. For the antigen, need to be FC free. So we can work with full-length antibody. The thing that we need to keep in mind is that we will only be able to visualize the Fab that is interacting with the antigen. And everything else that belongs to the antibody, like the second Fab and the FC, will be completely washed out, because I don't know if you remember the dancing antibody that I showed at the beginning. That is what antibodies actually do. And so we center, we focus, we align on one, and everything else will become very disordered, and basically we won't be able to see. Having or not having the full length antibody may affect the vitrification conditions, or may affect the way the particles sit on ice. So sometimes can be useful because it avoids the for example, preferred orientation that the complex might have. Sometimes it really interferes with the vitrification itself, or maybe forms aggregates or extended chains that we don't want to see. So it's a guessing game, basically. We like to start with a Fab, but we can definitely work with the entire antibody if present. How much data is produced for a single case test? Is it gigabytes or terabytes? It's a terabyte, more or less. So a data collection on our glaciers, if we do an overnight data collection, we go between 0.5 and 1 terabyte of data, more or less. So that's the thing. Is there a limit to the number of antibodies can be visualized at the same time bound to antigen? No, as long as they bind in different places. So if they are not competitive and there is enough surface of the antigen to bind 10 antibodies, we can visualize 10 antibodies. If they are competitive, of course, it doesn't work, right? Because one binds and then the second one will remove the first one. So no, it's really not a problem. It can be actually a good thing because it can reduce the preferred orientation if you have different things that bind in different positions. It definitely increases the mass, which makes visualizing the particles and picking them and aligning much easier for our process, our workflow. Epitope mapping is an application provided by the high-throughput SPR service companies. What do you think about it? I think it's a very good way to go. Again, it depends on the questions that you're asking and then the amount of details that you can have. I think it's definitely probably faster and cheaper than cryo-EM, but it will never give you the really atomic details that you're looking at. And so if you need just to have a general visualization of the area where the antibody binds, then I think it's actually better to use techniques that are higher throughput and less expensive than cryo-EM. But if you need to have the specific interaction between an arginine on your antigen and glutamic acid on the antibody, you might need to go to higher resolution. I always say cryo-EM and X-ray is always a complementary technique. All of these techniques that are available for solving the problems can be used isolated or together in a complementary way to just try to get to the answer that we need, the quickest way that we can get to the answer, basically. So it's not that one is better than the other. It depends on what we need. Can you map polyclonal epitopes? Yes and no. Again, it's a question of averaging. And so we can probably have, and since we are trying to put together things that at this point will be different, we will probably be able to get like an overall low resolution map indication of where these things bind. We won't be able to really assess with just one cryo-EM experiment atomic details. It will need to be done on a specific, on an antibody by antibody case. How successful are you with full map instead of just Fab, and what are the challenges? So we have used both. We'll use mostly Fab. I personally have worked with both. As I said before, sometimes I have cases in which the antibody, using the full length antibody, removed the preferred orientation problem that was happening with just the Fab, because the Fab has a very specific form and it tried to sit on ice in a very specific way. I had other cases in which, unfortunately, the full length antibody will form these chains, multiple chains, and then it became impossible to use the data for a single particle analysis. So and those are basically the two extremes, right? And a priori, I can never say, yes, we should use it, or no, we can't. It's something that we need to visualize and understand when we initially start working with our system. Do you frequently observe orientations bias for antibody interaction that affect your projected timeline? Yes and no. Again, antibodies, so Fabs tend to sit on ice in very specific positions. And so the presence of absence of a preferred orientation is very often driven by the antigen. So if the antigen try to compensate this orientation, we end up with not preferred orientation at all. It does happen on, I would say, 50% of the cases. We do have ways in which we can address it. It will increase the timeline a little bit, probably because, so the way we will approach the experiment is we run what we call a starter pack, which it will be a full characterization of the complex at the beginning, including a three-dimensional, an overnight data collection and 2D classification and an initial map. If we see that there is a preferred orientation, then we will need another day on the microscope in order to address the preferred orientation. We have ways to do it, from using detergents to other additives to eventually, if nothing works, to just do a tilted data collection, but it will add a little bit of a lag on the determination. Have you been able to map linear epitopes? I am not sure I understand the question. So our cryo-EM is a three-dimensional system, produces three-dimensional results. So we can map, we can see the epitopes, but they will always be in three dimensions. So we can see the peptide, if that's the question, but it will always have the three-dimensional component to it, not just the linear sequence. But if you want to clarify a little bit better or follow up on email, I will happy to follow up on that. Do you need to run a sec for the grid prep? Not necessarily. Again, that it's depending on preferences. We have been very successful in just mixing and matching. So taking the antigen, mixing with antibodies and going on grids directly. The grids can be a little bit dirtier because you have a lot of leftover things that maybe don't combine very well. It could be useful to have a fully purified complex, but it's not something that it's required. Have you been able to work with unstructured antigens, taus, alpha-synuclein, etc.? So what we'll be able to see in that case, I don't think I have any specific example that we have done, but I've seen it. What we can see in that case is the Fab and whatever piece of the antigen is bound to the Fab, and we won't be able to see anything else. And so it basically becomes a very complicated process because at that point it will be just a Fab bound to a peptide. And if peptide is very small, then the entire system is well below the 80 kilodalton that we like. And so we can do it, but it will be a longer and more complex system. And it won't give information of the binding with respect to the full protein, to the full alpha-synuclein, or to the full tau that you're interested in. Do the cost estimate that you mentioned to the Krios data collection, will doing the X-ray structure be less expensive? So the cost estimate that we go for this antigen epitope mapping does not include the Krios. We can do it faster for that amount of money in less than 2 weeks, only because we do all data collection on the glaciers. And we will make the grids, do a data collection, and then do maybe another overnight data collection or as much data as necessary to get to where we want to go. So Krios is not contemplated into the standard epitope mapping. If there is a request, we can go on the Krios, but that will be an added cost to the epitope mapping package. And it could be seen as one of our standard structure determination processes, which go to a different way, to a different path. Will be an X-ray less expensive? Again, yes and no. It depends on money-wise, probably. Time-wise, it depends. If you don't get crystals, you can do X-rays, right? And the good thing about cryo-EM is that every time you put something on a grid, you do see something coming out of the grid. And so you're going to get some kind of information, either it's falling apart or I need to change the preferred orientation. If you don't get the grades, it's like if you don't get crystal, it's a yes/no answer, right? And so maybe the amount of money that you spend in the 8 months that lead up to crystallization, it's equivalent to the amount of money that you will spend for the 2 weeks on the OEM. It's very hard to say. Any experience resolving the interaction with a small molecule as a cofactor in the fab or in the sample? So yes, we solve small molecule protein structures routinely. We solve them at resolution that are usually able to get to atomic detail, so we can clearly see the interactions. We can see the small molecule bound to something that's bound to a fab. It's something that can definitely be done. Do you offer services for just glacial data collection? Yes. If the user already screened grid condition and want to analyze data on their own. Yes, this is part of the fact that we are extremely flexible and we have multiple point of entry and multiple point of exit in our services. So if you have the grids, they are already screened, we can just collect data for you, either on the glaciers or on the cryos, and we can just give back the data to you and you're responsible for your own data processing. Or we can come in as early or exit as late as you want. So that will be a discussion that we need to have at the beginning, but we do offer we offer the possibility, any kind of access that will be best suited to your needs, basically. Do you provide antibody purification digestion services? No, we don't. We have a very limited biochemistry lab. We might start considering having some digestion kits in the lab, but we have not done it yet. And so at the moment we ask the client to provide the system, the complex, or the components ready to go on the microscope. Very often these unstructured antigens have linear epitopes. I was wondering if you will be able to pinpoint these epitopes on the full-length antigen by a high-res tool. Yes, you will definitely be able to see them and identify them. If you have a high-resolution structure and you know what your epitope is, you can definitely be able to visualize it. Why it takes 2 weeks or more per case? Could you please break down the time and what the time is spent the most? Okay, so for the moment we receive the sample, we will spend a full day, so 24 hours, on the microscope. We will spend the entire day trying to get the optimal vitrification conditions for the sample. Then we will do an overnight data collection, which is usually carried out on our glaciers, and we will run CryoSpark Live in parallel. So by the morning, an initial set of 2D classes will be available, and that will be very informative with regard to, is the sample suitable? Are the 2D classes good enough? Do I see secondary structure elements? Then it will take the analyst a couple of days to actually do a clear, good processing of the data and be able to either generate optimal 2D classes, and then from the 2D classes to get what we call the initial map, which is a very low resolution map, no refinement involved at all. It's very informative for us to see if there are problems, like preferred orientation very often is visible in the 2D classes, but it's really confirmed by the three-dimensional reconstruction. At that point, if everything looks good and we are within the parameters of the package, it's going to take another couple of days to find suitable time to get back on the microscope and collect all the available data necessary to get to the high resolution structure that we need. So we know that in order to be successful on an epitope mapping reconstruction, we need data to at least 3.5 angstroms, or at least data that allow us to trace the chain at the interface. And so that might require another overnight on the microscope. So it takes a couple of days to find that time. And then the rest is the time that is needed to finalize the reports, to finalize the structures, the three-dimensional reconstructions and everything else. Which software you mostly use for data processing? We are a CryoSpark shop. We have a CryoSpark integrated in our process. So every time we start a data collection, we start CryoSpark Live. It's very useful. It's very fast. We do have other softwares like System or Relion. in-house that can be used in case there is a need to change the software, but we usually stay with CryoSpark for most of the time. Will be there substantial price difference for structure determination of oligoclonal Fab, multiple non-overlapping map on a single antigen versus a single monoclonal Fab antigen structure? No, so the price we quote is the price by, basically is the number of, not number of grids, but the type of samples that we use. So if you have one antigen with 3 different epitopes, with 3 different Fabs attached, or one antigen with one Fab, the cost is the same, because for us, those are 2 individual samples, and we will treat the multiclonal one as we will treat the single one. So there is no difference in price, absolutely. Not to the up to the 3D map, to the 3D map generation at least, which is where the epitope mapping ends. Okay, I think we are well over the half an hour of the seminar. I would like to thank you all very much for attending. I hope I answered most of the questions, if not all of them. If you have any other questions, if you want to know more about the service, if you want to talk to me or anybody in the group, please reach out to us through the website or the email address that is in the website itself. And I hope you all have a good day, afternoon, evening, and see you soon. Thank you.